Good afternoon, everyone. Now I think we are switching gears a little bit. Are there any immunologists in the room? I don't think so. Well, that also makes my life a bit easier because I don't think this is new for anyone. The immune system is very complicated to study, and for some reason I decided to take this mission upon myself and try to unravel a little bit of what happens to the human immune system in space. Ah, pardon. Okay. And I think we already got a little glimpse of what happens to the human body in space when we had the wonderful presentation from Natalia, if I'm not mistaken. Maybe correct me if I'm wrong. And she already uncovered a little bit of the myriad of disruptions that the human body suffers when astronauts go to space. Of course, to make research a bit easier, we need to narrow down the field. We are working on the immune system, and the immune system is, as you will see, comprised of many organs and cells. So we also narrowed it a bit down more and focused on one particular cell type. So without further ado, this will be the structure of this presentation. I will start immediately by asking you, and I think this is the ideal audience actually, to ask you to visualize this future, because I think it's the main goal of this conference, is that we are pushing this very hard and we are all sharing this common goal. We want to go to Mars. We want to go to Mars soon, and we want to perform a lot of research. But I would like to highlight that we need people. It's people that will go to Mars, and people will be performing these tasks. So knowing that the space is a very harsh environment, we need to be aware that there's a lot of research that needs to be conducted in order to allow us to better protect the health of the people that we are sending to Mars, even though they are very willing to go. We know they are willing to risk. There are people that probably will go on a one-way trip. They will be fine with it. But it's also ethical to be able to protect them as best as we can. So the space environment, and this is a term I like a lot, it's comprised of a myriad of physical stressors and also psychological stressors that have to do with what the body is exposed to, but also are comprised of what is called the secondary exposome effects, which have to do with the daily life of the astronaut, not only the radiation that they are exposed to, or the altered gravity, or the eventual psychological stress that they will develop, but also the exposome is also comprised of their interactions with the habitat, with each other, with the food, etc. So there is this concept of the space exposome. So it's a myriad of factors that interconnect and cause a disturbance in our normal bodily functions, let's say. Of course, we always focus on the three main factors of the space environment, which are the radiation, the altered gravity fields, and the psychological stress. So to start with the radiation, we know that it's a very complex spectrum. So it's not only the radiation that astronauts will be exposed to that comes from our sun, but it's also the galactic cosmic rays. And here it's actually one of the factors of major concern in terms of radiation exposure to the human body, because there is a lot of uncertainty about these exposures or the effects of these exposures. And indeed, it is known that there are several factors or diseases that can arise due to these exposures. There is an increased potential cancer risk. This is what we know from radiation exposures. There is a very big uncertainty here because we are not able to know what happens in a real space scenario, because we are not going to send people just to test it. There is also altered neurological, cardiovascular, pulmonary, development of cataracts, and to no surprise, considering the theme of this presentation, there is an alteration in the immune system that's due to this radiation exposure. When we talk about altered gravity, the main concern is normally microgravity. We know that it causes a lot of disruptions in the normal status of every bodily system. But I would also like to highlight that it's also important to understand what partial gravity levels does. If the partial gravity level, for example, of the planet of Mars is enough that we don't see the effects that we see with microgravity, is there a threshold? Are we exposed to a gravity level that if we go below that, then yes, we develop these alterations, but there's a threshold. So these are questions that are still open. And again, a lot of different organs and systems are affected by it, and the immune system is no exception. And then we have the stress, and this is probably one of the most complex to try to understand where these stress factors are coming from. But what is known is that there is a physiological response to this psychological stress. And one of these physiological responses is the alteration of our pituitary adrenal gland axis. I'm probably saying this wrong, but it's, yeah. And what this mainly does in the physiological response of this is we have an altered level of cortisol, which is one of the most known stress hormones. So there is an increase of the levels of this hormone in the blood. This has been found in astronauts, in the urine. So we know that this exposure to this environment causes this physiological response. And of course, cortisol is actually one of the most studied hormones that affects the immune system. When we have an inflammatory response, for example, an autoimmune disease, it's one of the drugs that is administered to counteract, to kind of suppress this immune response, is hydrocortisone or a corticoid, so it has to do with cortisol. Cortisol is a known immunosuppressor. So just briefly about the immune system, as I said, super complicated, but I think to also give you an idea, it can be organized in terms of the organs that comprise the immune system, and it can also be organized in terms of the cells that comprise the immune system. Well, don't worry, we don't have to know all of them. As I told you, I focus on T cells, so we are only going to talk about T cells today. But I think it's also interesting to know that there are several levels of protection in our immune system. We have the first barrier, which is composed of physical and chemical barriers against external pathogens. Then we transition to a cellular immunity, and these two together comprise our innate immunity. This is the one that is activated immediately when we face a pathogen or a stress. And then if this is successful, we create memory, and this is what we now see as adaptive immunity. This is what happens when you get a vaccination. You are priming your adaptive immune system to recognize the pathogen that you were vaccinated against. So, as I said, immunologists like to complicate. I think you can see through this image. But I find this very interesting. Of course, we are not all here interested in all of these myriad of pathways. Not even I am interested in all of them. But this is a very interesting recent review, I think it's last month. And it coined a new term for this field of research, astroimmunology. And basically, this is a model immune cell. And here they try to summarize what happens from all of the exposure stressors that the immune cells are exposed to in space. So let's see, we have here, as I mentioned, cosmic radiation, microgravity, the physiological stress, altered the microbiome because it's a hostile environment, and we are all carriers. We contain multitudes, so we are all carriers of several of our own microbiome, and we are going to share it with other astronauts when we are sharing the habitat. So there is an altered microbiome disrupted by this interaction, and also disrupted by the stressors that space ensues. And then from these exposures, we have a myriad of alterations. As you can imagine, we are altering the way the cell functions. And one of the interesting ones, for example, is the exposure to this microgravity or this altered gravity disrupts the cellular skeleton. And this disrupting the cellular skeleton actually has a very interesting consequence. We are changing the way the genes are expressed. The genome is compacted in the chromosomes, and if we are altering the way the physical properties of this compact myriad of chromosomes, you are changing the way that the proteins that are responsible for the transcription of the genes can access those genes. So this is one important, for example, factor. And in the immune system, it seems that this is also one of the reasons why we see a dysfunction of the immune system in space. So this whole picture can be summarized by this table, also in the picture. So basically what we see is that we have down regulation of IFN cytokines. These are interferon cytokines that are very important in this earlier phase of the immune system, the innate immune system. They are responsible for the response when a pathogen is detected. These are the cytokines that are released by the cells to call on the other cells that are responsible for the clearance of this. And then there is a whole also panoply of cytokines that are actually pro-inflammatory. So there is, on one hand, a disruption that is anti-inflammatory, but also one that is pro-inflammatory. So we are still trying to figure out why this happens. And we see that we are diminishing the capacity of the adaptive immune system to respond, and also the capacity of the innate immune system to clear pathogens. So there is a clear risk for an astronaut if they are facing any kind of infection, for example, since they are in a new environment where they have to react to the possible contamination, for example, of the habitat. And if they have their immune system that is less primed to be able to respond to this new infection, that could be problematic. So this is the main problem that we are trying to understand. And of course, some open questions that, like I mentioned. So this, as I showed in this scheme, it is the response to these several exposures, altered gravity, the stress, and the radiation. But what happens in space is that we are not able to understand where this effect is coming from. Is it coming from the radiation, or is it coming from the gravity, or from the stress? We don't know, because you are exposed to all of these main three at the same time. So this is one of the questions that we wanted to try to understand. Is there a combined synergy of all of these factors? Or is actually what we are seeing is the responsibility of one of them? So this was one of the open questions that we had and that we tried to do by using this and then studies in the laboratory with T cells. So we use T cells in the laboratory, and in the laboratory we need to simulate these space conditions because we were not able to send these cells, unfortunately, to space. And then we exposed these cells to these individual simulated space stressors to try to understand what each of them does and try to maybe unravel a mechanism. So, as I said, we use T cells and T cells because they are very important coordinators of the immune response. They are the ones that will dictate if the immune system will be pro-inflammatory or anti-inflammatory when the response is enough. The pathogen is cleared. They are also the ones that recognize this and say the rest of the cells, calm down, everything is fine. So they are very important in this response. And they produce cytokines that are pro-inflammatory or anti-inflammatory, and one of them in the pro-inflammatory part is IL-2. And just also to summarize, these are many of the ways that we can do the simulation of the space conditions on Earth. I don't need to read this all, also for the sake of time. I will just highlight that for us, we use the random positioning machine that allowed us to simulate then these altered gravity levels. We expose ourselves to space-relevant radiation In particle accelerators, we use heavy ions, which is part of the composition of galactic cosmic rays. And for stress, because we were working with cells in vitro, we used an analog of cortisol, basically exposing the cells to cortisol. So these are our model cells. These are a type of T cell. We wanted to test them in an activated state, meaning that we mimic a moment where the cells will be exposed to, for example, a pathogen, and they are activated. They need to release these pro-inflammatory cytokines. So this is what we did. And what we also tried to test, as I mentioned, was cortisol. We simulated the Mars gravity level, simulated moon gravity, also microgravity, and then two types of radiation. Carbon ions are part of the spectrum, but X-rays, because we always like to compare to a standard type of radiation, just to check the differences. And as I mentioned, we did it in different combinations because we wanted to isolate or try to isolate the effect of each of them. So, and this IL-2 cytokine, which is a pro-inflammatory cytokine I mentioned, was how we quantified how the cells responded to this, basically. So, this resulted in the publication. If you are interested in this, you can check it. There are more data in it, of course. So, here we see the levels of IL-2, of this pro-inflammatory cytokine. And when we have no radiation, we can see that exposing the cells to the Mars gravity reduces how much they release to the media. When we expose them to moon gravity as well, and when microgravity as well. We would expect that this will be linear. The higher the gravity, it will decrease. This appeared not to be the case, but I think also more data is needed for this. Interesting, when we expose the cells to radiation photons, we see that we decreased even more the levels of this cytokine, and Mars again showed to have the most impact in combination with photon radiation. But this was not the case when we expose the cells to carbon radiation, which is these heavy ions that are the same as the ones that we find in space. Mind you that the doses have nothing to do with the doses that we are exposed to in space, but we wanted to kind of do a proof of concept, let's say. And what was interesting for us is that from the moment we add cortisol to these cells, we already see an impact, even if there is no radiation. So it could indicate that maybe stress and the increased levels of cortisol are what is actually more hindering this pro-inflammatory capacity of these cells. But of course, further research is needed. On the course of my PhD, which is what made me do this research that I'm showing you today, we have more experiments. For the sake of time, I'm not going to show them all, but if you are interested, you can come to me. I can show you the papers or discuss it with you. But this was very interesting, and I wanted to show you here how also interesting that we can try to simulate the levels of gravity of the planetary bodies that we are now really looking forward to go to. But we want to go to Mars, and it's important that we try to understand as well if we can already use countermeasures that can help the astronauts. Before countermeasures, what is important is to measure, actually, what is happening in space, what is happening to the immune system. So it's interesting to understand and develop methods that work in space, in microgravity or in altered gravity levels. So there is a lot of effort in terms of technology to be able to conduct these measurements in the mission. Not just like, for example, what happens in most of the cases now is that you are measured before you go to space. You take samples during, the samples are maybe returned, and then you take measurements after you return, but not during the mission. So it's important to try to develop a best way to analyze if there is something happening in the moment with astronauts in terms of their immune system. For example, measuring the levels of cortisol in urine or in saliva, which are also a good candidate for measuring these alterations. And then after understanding what is happening, there are also countermeasures that are already in place. For example, we know that exercise is one of the best countermeasures for not just the immune system dysfunction, but for many others, like skeletal muscle, bone loss, etc. But we also, if we are able to use the measurements, we are able to better adapt the countermeasure to what is happening. For example, in terms of nutrient intake, we know that this astronaut has a problem. Maybe we can adapt the diet of that astronaut, and the other one doesn't need it, for example. So these are still areas that are under heavy investigation for the people that are working in this, because it's very important. And also in this slide, there's a lot of details. I have to be honest, I'm not the expert on most of them, but there are already countermeasures in place to what we know can happen, namely dietary, for example, with probiotics, with the pharmacological interventions that are also already known to be able to counteract some of the problems that astronauts find in space. And just briefly also to tell you that there are a lot of research being, as I mentioned, being made in order to be able to do this in flight. And then personalized approach. I'm finishing, I promise. So basically my take-home message is, if you are a biologist, radiobiologist like myself, you would be keen to integrate the immune system if you are studying other systems that also are disrupted with space, for example, muscle, bone. Integrate the immune system, because it will be relevant and it changes your approach. But if you are, like we are here, we talked a lot of analogs and space analogs, integrate also studies about what isolation and confinement does to your immune system. And this can easily or relatively easily be done if you need cooperations. I know for sure there are a lot of scientists willing to be able to cooperate with the organizers of these analog missions to be able to take out as much data as we can from the analog astronauts in relation to the immune system. So basically, we need you to be able to also understand that this is important to study in order to make this a reality and protect the little fella that we will send to Mars. Thank you. Of course, thank you to the Belgian Mars Society for allowing me to come here. And this work would not be here if it wasn't for the people that helped me throughout my PhD journey. So thank you for the opportunity.
Yes, not a lot of questions, please, but yes.
A comment and a question.
Yes.
Interestingly, we see a lot of the stressors that happen in long-duration spaceflight also in analog missions. I mean, cortisol levels elevated is something we see in almost all the missions we are measuring. We found even, you might be aware of telomere shortening of astronauts in long-duration spaceflight. In a recent study for MIT-24, we were demonstrating this in analog missions as well. We saw telomere shortenings within 3 weeks roughly. It's significant and the borderline threshold, but it's amazing to see this cellular change. What I'm interested in here comes the question is, when you have the cortisol level changed, how much do you attribute this to 0G versus just the elevated stress level? You cannot differentiate that, right?
So we treated, in our experiments, we also treated the cells with cortisol, the cortisol elevation. So the cortisol elevation has many factors. Being in micro G can be one of them, because there is a kidney also function disruption, and the adrenal glands are also there. There can be an imbalance in the fluid distribution, for example, and the availability of cortisol. But this is debatable because we have still a blood system that has a pressure and that shouldn't be as problematic. However, cortisol can also be elevated just because they are stressed that they are in micro G. But they cannot see properly, suns. It's not, there is not one thing that will happen that causes elevation of cortisol. However, when you say that you see this in analog missions, then maybe it's the stress and not micro G. So I think, yeah, it's a very interesting question, but thank you.
Yes.
From the nutrition point of view, could you mention some of the solutions you are investigating in this research? And also, from this side, what could be the possible application on terrestrial medicine for people who have same problems with
Yes.
the nervous system and T cells?
So, to be perfectly honest, I'm not really aware of specific countermeasures in terms of nutrition. I know that there are a lot of dietary supplementation that you can do that can help in other ways, and that might, how do you say, indirectly affect how the immune system responds, because the immune system alterations are also not caused by one problem, which is also another added factor of complication. But probiotics are one of them. So if you try to maintain your gut microbiome, it is it's already established that the compromising the gut microbiome is a major factor in the several alterations, namely immune system alterations. So this could be, for example, one way. I think for earth applications, for people that suffer from immune system disorders, if we try to, from this, go to mechanistic studies, for example, studying genetically what happens to cells, what are the triggers in a timeline of exposure, for example, what is the beginning of this alteration, what is the mechanism that is activated, maybe we can then use this knowledge to help people on Earth by counteracting that mechanism in the beginning of the disease, for example. So, but yeah, I don't know, more questions?
Thank you. Now I think we don't have time for questions because we're already very late, more than 15 minutes late. So thank you very much.